Okay. All right. Going back again here. Okay. Sorry about that. All right. So now I'm recording. Nobody really missed anything anyway. Okay. For lab, I don't know if anyone watched my it's probably a pretty sorry demo, but I think I got everything covered in my office there. So these are videos, right? The metric and microscope lab. And so there's a part 1 and a part 2. And basically, remember, they correspond to this metric and microscope lab handout. So that's due on Thursday, the completed lab handout. I think I pretty much explained everything in the video. So just let me know if there's questions. And remember, so the assignment is here, right? To basically complete that lab handout, that PDF, submit it here. And remember, there's also another assignment, which is just some practice conversions. Make sure both of those are submitted by Thursday. All right, any questions about the lab? All right, so while we're on that topic, the upcoming labs for modules 4 through 6, the first one here is the chemical composition of cells. I will be recording that tomorrow, so that'll be up. And then there's a cell structure and function lab. Part of it will just be like kind of going through sort of I don't like to say the word worksheet, but a worksheet where you're just labeling cell diagrams and just writing down basic function. And then another part of it will be actually a video from this online sort of, there's this online journal that is all videos, and it's mostly used for techniques and stuff like that, but now they have a lot of lab demos on there. And so I will have a, basically like a, sort of like a little lab report kind of thing already made up for you guys with some questions, and you'll be completing that and it'll correspond to that video. Okay, again, will all be explained in the videos that I post, but I just wanted to give you guys a little bit of a heads up with that too. So then that'll be due that following Thursday. All right, so that's kind of the next, those are the labs that are in these next set of modules, right? Which is technically for this week. So like I said, it'll be a chemical composition of cells, meaning like doing a test. How do you test for the presence of sugars, right? Test for the presence of starch, tests for the presence of protein. So there are these chemical tests that you can do in the lab to detect the presence of them. So that's the first part. And then, like I said, parts of the cell. And then also there's a lab on like diffusion and osmosis. That would be a video. Okay. So that's upcoming. And then it's chapters 4 through 6. So I'll be posting the recordings for the chapters as well. All right. Any questions, guys? Okay. The first exam is on chapters 1 through 4. So again, the dates are there. I'll remind you again, saying it's available. You'll then be able to see exam 1. It'll look like one of the quizzes. Okay. But obviously it's going to be a little more lengthy. Click on it. It'll have the due date, have the time you have to complete it and all that good stuff. Everything will be in there. You just don't see it yet. Okay. So what I wanted to do, like I said, is to go over the homework questions. So I don't know if you have the homework questions available, yours in front of you, that would be great, because I'm going to ask, sort of volunteers to at least give me an answer or two so that it's not just me talking. I'm not just ever going to just post the answers because that's not really that helpful. I do have some of these already, like the answers written in. So if somebody has a question about something, if something wasn't clear and you need that clarification, let me know. But in general, I don't just post answers because then people just copy. And so it's not really worth it. So it's up to you to have your homework out right now for us to go over it. And then we'll go from there. Okay. And also, like I said, some people still haven't submitted the homework. So again, if I just post the answers, people can copy paste. So that's why I want it's nice to have the homework done before we go over it. All right. So with that said, let's start with chapter 1 here. And I do have the lectures handy. So if I need to pull up and clarify something, I will. So number 1 asks about the common characteristics of life. Super straightforward. This chapter is super basic. Some of these things you probably already know. You don't need to read in the chapter. So what'd you guys put down, or give me a characteristic of life, somebody?
Living organisms are organized.
Thank you. Living organisms are organized. Good. What's another one? Somebody else. What's another characteristic of life? Anyone. I don't want to call on you guys.
It requires material and energy.
Okay, good. Requires materials and energy. All right, good. What's another one? Somebody else started talking.
Composed of cells.
Composed of cells. Okay. What else? Melissa, did you have one? Or you already said one?
Yeah, I already said one.
All right, you're already off that. So the living things are organized in sort of the composed of cells kind of goes within there, right? And then, sorry, the next question is that. So let's see, what else we got? Requires materials and energy. Somebody already said that. Homeostasis, right? Living systems maintain homeostasis. So how would you describe homeostasis, or what's kind of like one or two words to describe homeostasis? Like a quick definition. Kind of says it there on my slide there, but what would you say? Like steady state, right? Just maintenance of steady state. Another one, living organisms respond to stimuli. Okay, you have to be able to respond to stimuli in your environment in order to function, right? And to survive, and to be alive. What else we got here? Oh, living organisms reproduce and develop. I'm sure we all agree that if you can't reproduce and develop, you're not going to be too successful. Living organisms have adaptations. I think that's the last one, right? Yeah. All right. So those are, I think we listed 6 of them, right? Number 6, and remember they're numbered in the slide, in the slides, just like we went over here. In terms of, let's go to question 3, just because that sort of corresponds to where we are in the lecture there. So it says, how does natural selection result in adaptation to the environment? So somebody, how would you sort of relate natural selection and adaptations, I guess, is sort of kind of what I'm asking. What'd you put for that?
I said that those with traits that are best fitted to survive in an environment are more apt to be passed down than those that lack these traits. Therefore, organisms are going to adapt to the environment in order to survive.
Perfect. Yeah, exactly. Right. The other, I guess, point that I always try to make here is that, remember when we talk about, selection or natural selection, that those traits that those animals have, right, are just, they're there sort of by chance, right? They're there because of genetic changes or mutations that occur. But then if that, trait gives them an advantage, right, then it's going to make them more fit for that environment and more, they're going to produce more offspring, right? And they're going to be more successful, just like Alicia said, right? So just remember, when we talk about natural selection, it's the environment selecting for those traits that are most favorable, okay? The traits, though, are dictated by the genes, right? By the DNA. And that what's there or what's present there is really just by chance. But again, if that makes that organism more suited for the environment, then you're going to see those traits, right, occur over and over again and get passed down to generation to generation. All right. And so number 2, right, what those levels of biological organization go along with what Eloise said, right, when you talked about, or you mentioned cells, right, that organisms are composed of cells. And so when we talk about the different levels here, we're going from most simple, right, which here is the atom. And what's next, guys, after atom?
Molecule.
Good, molecule, right? All right, and then what about after molecule?
Cell.
Good, cells. All right, and so remember, in terms of this, we should know these levels, and you should really know, on the left-hand side of the slide, there's a little basic definition. Should be familiar with that too, or a description, I should say. What's next after cell?
Tissue.
Tissue. All right, good. All right, so our example here is nervous tissue. And I believe that was leaf tissue. And then, oh, sorry, I flipped too soon. What's after tissue?
Organ.
organ, right? So when we're on the animal side there, our example of tissue was nervous tissue. And now if we go up one level, right, further, in complexity, now we're talking about the brain, which is an organ, right? And in the plant, the leaf, right, which is an organ of the plant. What's next after organ?
Organ system.
Good, organ system. And so if we're talking about the brain, what organ system is the brain part of?
The
What
nervous,
organ- Say it again. Sorry.
the nervous system.
Right, nervous system. Right, and then for the, oops, what the heck did I do? Oh, that's because I don't have it in slideshow. For the plants, right, shoot system is for the leaves. All right, so what's next after organ system? What do you got next?
Organism.
Good, organism, right? Why are you not changing? All right, so now we got organism. And then if we go up in complexity from organism, what's next?
Population.
Good, population. So remember, population are organisms of the same species, right? So this is all one species that live in a particular area. After population, where are we at next?
Community.
Okay, good. Community. And so now you're bringing in different populations, right? So different species interacting with each other, but they're all in a particular area. All right, next. Make it bigger. Right, now we have ecosystem because now we're basically considering the physical environment as well. Right, and then why doesn't my arrows work? And then lastly, sort of most complex, biosphere, because now we're talking about regions of the Earth's crust, right? The atmosphere, that basically all those living things are a part of. All right, so we should be familiar with all of these things. So like I said, that chapter 1 is really just introductory. Probably a lot of the stuff you've heard of before. It's just, and that's what it's meant to be. It's not meant to be anything crazy, just an introductory chapter. Yeah, so we talked a little bit more about natural selection, right, and adaptation. We already sort of talked about that. Does anyone have any questions, particularly pertaining to chapter 1, that you want me to clarify or review or anything like that? No? Okay. All right, so let's move on to chapter 2. So chapter 2 was really just, some chemistry, basic, very, very basic chemistry, right, to sort of set up for our course. So what is an isotope? Okay, then it asks about a radioactive isotope, and then 2 uses of radioactivity in science and medicine. So does somebody want to tell us, I guess, what you put for in terms of defining what an isotope is? How'd you define an isotope?
I said that an isotope is like atoms of the same element, but they differ in the number of neutrons, so they have different atomic masses.
Good. So, right, they differ in the number of neutrons, right? So they have different atomic masses, and some of these will spontaneously decay, right? And then if they're radioactive, they will then give off energy, right? So when we say it's radioactive, so not necessarily every type of isotope is a radioactive isotope, but many of them are, right? So I think that was the second part of the question, asked about radioactive isotopes. So that's going to be an isotope that gives off energy, right? And some can be harmful and some can be helpful. And so the next part of the question asked about what are some of the ways that we use isotopes, radioactive isotopes in particular, in science and medicine. So somebody want to give me an example?
So for the low levels of radiation, like medical imaging, if you consume like what it's detecting, then you can get a clear image of like the internal organ you're looking at.
Good, very good, right. So those are low levels. And so, right, something is tagged, right? Some molecule, maybe glucose, right? Or iodine is tagged with a radioactive isotope. You then inject the patient with it, not ingest it, usually it's injected. And then that, whatever that molecule in particular is going to go to a particular organ. So the two examples we gave, right, were imaging the thyroid, okay, using radioactive iodine, and then imaging the brain using radioactive glucose, right? So what do you call this in panel B, this image of the brain where you basically inject radioactive glucose? It's called a PET scan, right? So, so again, it's really just showing you, it's actually showing you sort of activity, right? So you could potentially pick up areas where there's little or no activity due to damage, right? It could be due to damage of a stroke, you know, caused by a stroke or something else. So those are really powerful techniques, right? And so using low levels. All right, so what about high levels? What's the one example, or I gave two examples of using high levels of radiation.
You can use radiation therapy to kill off tumors.
Good, radiation therapy to kill off tumors. And so again, remember, this has to be super focused, right? If you exposed a patient to high levels of radiation everywhere, well, you're going to start killing off cells that you need, right? It's going to kill off any sort of rapidly dividing cell. But you can focus that, right? And so this is showing you an area, obviously focusing into a tumor on the brain, and help basically destroy those cancer cells in that one really focused area, right? And so they use lasers to do that. And then panel A, I always sort of, I don't know if I feel like I get disturbed when I see that panel A thing, right? But you know, you can use high levels of radiation, expose fruit to it, because it will then kill off any fungi or bacteria that are on the outside of that fruit that then will cause it to decay faster, right? And so high levels of radiation can be used that way as well. All right. Okay, good. So let's look at that next question. Okay, so explain why water is a polar molecule. What does the polarity and shape of water have to do with its ability to form hydrogen bonds, right? So kind of a long question, but I think this sort of goes all hand in hand. So let me, well, one, what does it mean to be a polar molecule? And that sort of, you can kind of start with that definition to sort of explain that first part of the question. What does it mean if a molecule is polar? So here, let me do this. So here, if we look, oh, what did I do? Sorry, wait. So if you guys look at this slide here, right, we were talking about covalent bonds. You know, let's back up just a few slides. Part of this chapter, you know, we talked a lot about electrons, right? Electrons in the valence shell, right? Which is that outermost shell. And the reason why we're talking about those valence electrons and the numbers of them is because that dictates how that element behaves, or how that atom behaves chemically, right? And so for the most part, we'll focus on the number of electrons in the valence shell. One of the types of bonds that forms between atoms are covalent bonds, right? And so if you guys look at these examples that are here, right, of hydrogen gas, oxygen gas, and methane, are those molecules polar or non-polar? What do you think about hydrogen gas here? Is that polar or non-polar? Yes or no? What do you think? Polar? Yes or no?
No.
Correct. They're not polar. Why?
Their electrons are shared equally.
Correct. Their electrons are shared equally. And even if you just look at the structural formula, right, that basically denotes that, or shows that, right? Those hydrogens, you know, the bond there, essentially those electrons are shared equally. And so the molecule is non-polar. There's no difference Okay, it's sort of symmetrical, right? So now if we look and we talk about a polar covalent bond, those electrons are not shared equally, right? And so our example is, I guess we'll look at that there, is water, right? So if this was polar and we were going to write the structural formula as it being polar, you would see like it would be that these hydrogens were exactly in line with the oxygen, right? But that's not true. This is a polar molecule, right? Because of unequal sharing of electrons. And then what happens, right? You end up with oxygen having a partially negative charge and the hydrogens having a partially positive charge, right? And so then that leads to that next part, or the last part of the question, that asks about the formation of hydrogen bonds, right? Sorry, let me just, I think I just asked you, yeah, why does the polarity in the shape, sorry, what does the polarity and shape of water have to do with its ability to form hydrogen bonds, right? So if we go back and we look at that, you know, why does it form hydrogen bonds, or why is it able to form hydrogen bonds? What's sort of dictating that, I guess, or allowing that to happen? It's what I just talked about.
I said that multiple hydrogen bonds can be formed because the oxygen is partially negative and the hydrogen is partially positive.
Correct. And so that partially negative oxygen is going to attract other positive molecules, right? It could be an ion, a positive ion, but it also can just be other hydrogens from other water molecules, right? So anything, so you're always going to get this sort of attraction between positive and negative, right? And so if you think about a whole bunch of water molecules together, hydrogen bonds form between them, and right, it just simply is that interaction between the partially negative oxygen and those partially positive hydrogens, okay? I mean, it kind of seems like a simple thing, but as you kind of go, as I went through that lecture and we talked more about water itself and why it's so important as a biological molecule, I hope you sort of gathered that really everything, or all of the properties of water, have to do with those hydrogen bonds that form, and really the interactions that are caused because of water being a polar molecule, right? And that partially negative oxygen and those partially positive hydrogens. So yeah, so again, if you want me to go over anything else from that, I can. Yeah, I guess I didn't really ask you more about that, but you should be familiar with the properties of water, right? And again, just understanding that all of these things are possible because of that, you know, water and its structure and the fact that it's a polar molecule, okay? And those things that we just talked about before, right? So anyway, make sure you guys are familiar with the properties of water. I mean, again, I'm on the exam, I really get away from the essays just saying, hey, list the properties of water, right? It would probably be something involving one of these properties, and then just asking you maybe a little bit more kind of to try to get at your understanding of that, okay? All right, those two terms that are up there now, hydrophilic and hydrophobic, we should be familiar with those as well. All right, guys, so let me now go to that last question. Contrast atomic number and mass number, okay? So that's a super basic question. We go back to the beginning, right, of the lecture. Okay, so if we're talking about atomic number, it's equal to the number of protons, right, in an atom, and the mass number is the number of protons and neutrons. How come we don't, how come electrons aren't part of this? We don't add the mass of the electrons.
Isn't the mass of the electrons equal to the protons?
No, the mass of the electrons is actually negligible, right? So the electrons really don't have a mass, and so that's why you don't take them into account when you're talking about the mass. But if you think about the charge, right, electrons are negatively charged, right, and protons are positively charged. So in terms of that, they'll sort of, I guess, kind of balance each other out, right? And then it's the electrons that we really talk about, because the electrons, even though they're not part of the mass, right? So if you look here, right, you can see the charge, but if you look at the atomic mass unit, you can see electrons, it's 0, okay? So that's why you don't take them into account when you're calculating atomic mass. But again, the charge is super important. And again, the electrons dictate how those atoms behave chemically, right? So that's why we talk about them a lot. But if you're talking about atomic number and atomic mass, right? Atomic number is the number of protons, and the mass number, or the atomic mass, is the number of protons and neutrons. Does that make sense to everyone? Does anyone have a question about that? No? Okay. All right. So finally, if we move on to chapter 3, okay, chapter 3, actually I asked you a lot of questions, I guess, in chapter 3, some of them short answers, right? Chapter 3, now, we start getting a little more complex, right? So talking about biomolecules. So I guess we can just start off. Tell me what the 4 classes of biomolecules are. Somebody shout those out, or you can give one example or all 4. What are the 4 classes? Give me one. What's one type of biomolecule?
Carbs, lipids, nucleic acids, and protein.
Good. Carbohydrates, lipids, nucleic acids, and proteins, right? Those are the 4 classes of biomolecules, okay? And so this chapter basically starts off by, in general, talking about organic molecules, right? And then reactions that are used to synthesize biomolecules, and also reactions that are used to break down biomolecules, okay? And then basically I talked about every single class of biomolecules. So the first question in this chapter 3 homework says, describe the synthesis and the degradation of one type of biomolecule, right? So you could have talked about proteins, you could have used carbohydrates as your example, whatever you wanted to do. So and again, the sort of general reactions that we talked about are these here, right? And so remember that these reactions apply to all of these biomolecules, okay? So when you're talking about synthesis, what type of reaction is this up here? Says it, but I mean, dehydration, right? So I'll ask you this, why is it called dehydration?
Because
why is it called a dehydration reaction?
Because water is being removed in the process.
Good. Water's being removed in the process. You can see that here. And when we talk about synthesis, we're talking about synthesizing a biomolecule, right? Joining together 2 monomers, or joining together 2 subunits, right, to form a more complex molecule. And so you could think about this as being 2 amino acids. If these were 2 amino acids, what biomolecule would we be talking about? Amino acids are the subunit or the monomer of what type of biomolecule?
Protein.
Proteins, good. Okay, so your example could have been, right, 2 amino acids, dehydration reaction, they're joined together, peptide bond here to form a protein, right? If we then talk about degradation, right, or breakdown, okay, you can go with the same thing, same example we were just talking about. Here's your protein, really, here's your polypeptide, not really protein. And a hydration reaction is going to be used to basically break the bond here, right? To break these back up into those 2 monomers. So you have a polypeptide, right? And then basically this hydration reaction, why is it called hydration? Because water is being put in, and now you have it back to those 2 amino acids. And remember that this is oversimplified, and this is showing you what's happening at each spot, right? At each bond that's formed between those monomers. You could also, think about this in the context of carbohydrates, right? So if we talk about or think about more complex carbohydrates like starch, it's composed of glucose, right? So it's glucose subunits that are joined together. All right, so let me just see what else I asked you guys in those homework questions. All right, so I said name 2 types of polysaccharides and describe their function. So I just gave you one, right? Starch, okay, is a polysaccharide, a complex carbohydrate. And so what about starch? What would we say the function of starch is?
Provides energy storage in plants.
Yeah, it's energy storage in plants, okay? So again, it's made up of glucose. So glucose is stored in the plant cell, in the form of starch. All right, so along those same lines, but in animal cells, what is the energy storage polysaccharide in animal cells? Oh, I just
Glycogen.
have it up there now. Yeah, glycogen. Okay, so again, same thing. It's made up of starch. So if your blood sugar levels drop, right, there's going to be hormones that are sent out that would then trigger your body to break down glycogen, right? So that then there's more glucose available. Okay, so it's again just a storage molecule. You could have also, for that question, listed cellulose, which is a structural polysaccharide, right? Or chitin, a structural polysaccharide. Also, peptidoglycan is a structural one as well. Okay, so those are all examples of polysaccharides or complex carbohydrates, however you want to refer to them. All right, number 3, describe the difference between an unsaturated fat and a saturated fat. So what's one, just kind of right off the bat, one major difference between saturated and unsaturated? Think about the bonds that are in the molecule.
Unsaturated fats have a double bond and saturated fats don't.
Good. Unsaturated fats have double bonds, 1 or more double bonds. Saturated fats do not. And then what happens as a result of that, right? If you look at those fatty acids, I pull up this picture here, right? You can see here, here's our unsaturated fat, things like corn oil, olive oil, right? Have double bonds. And so what happens is this kind of forms kinks in the chains of these fatty acids. And so there's more space in that molecule, okay? So it creates space. Here in the saturated fat, so things like butter, right, that are solid at room temperature, you can see there are no double bonds, right? And that these fatty acids are closely packed together, okay? And so the double bonds basically affect the structure of the molecule, right? Which then affects the chemical sort of behavior of the molecule, okay? So you should be able to identify these. So if I showed you, remember, nobody's drawing structures or anything like that, but I mean, if I showed you this and I said, is this an unsaturated fat or a saturated fat? We should be able to point out, we should know that, right? Just because of the presence of the double bond. I'm not going to ask you what exact type it is or anything like that, right? But we should be able to identify that. I mean, in same way, if I showed it to you like this, we should be able to identify that this is the unsaturated and this is the saturated, okay? Any questions about that, guys? I won't just let me out. All right, so let's just check our last couple questions and then we're basically done. Unless you guys have more that you want me to go over. Oh, what are the monomers that make up proteins? You guys already answered that, right? Amino acids. What are the monomers that make up nucleic acids? So what are nucleic? Well, so one, what are our examples of nucleic acids? Two main examples of nucleic acids. There's another one too, but DNA and RNA, right? And so what are the monomers that make up DNA and RNA? What do we call them?
Nucleotides.
Nucleotides, good. And so if we want to name a few nucleotides, right? And we know that they're slightly different between DNA and RNA, right? So DNA, adenine, guanine, cytosine, and thymine. And remember, in RNA, there's no thymine, it's uracil instead, okay? And then, you know, I went through sort of just the basic structure of DNA and RNA. We talk about that more again, you know, when we talk about replication and we talk about transcription and things like that as well. Let's see if there's couple other things. Just to point out, just a reminder, right? Pyridamines versus purines. So the purines are adenine and guanine, the pyridamines, cytosine, thymine, and uracil. a pyridamine always pairs with a purine, right? And so if we think of cytosine, what does cytosine always pair with? Right? Cytosine C always pairs with which one over here? A or G?
G.
G, right? And if we're talking about thymine, right? T pairs with A, adenine, okay? Uracil pairs with A as well. All right, I don't know. I mean, there's a lot more information, obviously, in the chapter than the homework questions. I don't hit everything in there. But does anyone have any other questions about any of the chapters? Chapter 1, 2, and maybe in particular chapter 3, because that's what we're just kind of focusing on. That's probably up to this point the sort of most, I guess, involved chapter so far. Any questions about chapter 3, guys?
Can we go over ATP a little bit?
Yeah, sure. Actually, that's funny because I was going to mention that, but let's quickly go over that. So, okay. So for ATP, right, it's a nucleotide, okay? It's composed of adenine, ribose, and then 3 phosphates, right? Hence the name TP, adenosine triphosphate, okay? You know, in general, ATP is the sort of energy molecule of the cell, okay? And basically any reaction or chemical reaction that's going to need energy, it's for the most part going to get it from ATP, meaning once the bond between the 2 phosphates here is cleaved, right? So if we're talking about adenosine triphosphate, ATP, you have 3 phosphates on it, right? So if within these bonds, there's super high energy. So lots of potential energy stored in this molecule, yeah? So if you cleave this bond here, it's going to liberate energy, right? And so that's what this is showing you here. So now you end up with ADP, right? Adenosine diphosphate and a free phosphate. But what you're really concerned about, or what you really want, is the energy that's liberated here. So basically, this sort of reaction of cleaving off that phosphate group gets coupled to other reactions within the cell, so that another reaction, say like synthesis of a protein that requires energy, can basically utilize the energy that's derived from the breakdown of ATP. Okay, now it is kind of complex, and it's something that I talk about a lot more in cell bio. For our purposes in fundamentals, we kind of leave it at, let's know that ATP is a energy molecule, or the energy molecule of the cell, and the way that the cell is going to get energy from that ATP molecule is basically by cleaving one of those phosphate bonds. Does that help?
Yes, perfect. Thank you.
Yep, you got it. Other questions, guys? Ooh, what the heck happened here? Sorry, I got a weird thing on there. Any other questions pertaining to chapter 3? No? All right, so let me just, I don't know why I did that. Let me just reshare my screen again. I'm just gonna go back to Moodle, just kind of hit those high points again, remind you, but otherwise we're finished. So the one big thing is that your lab, that metric system and microscope lab, are due on Thursday. Okay, they're due by 11 a.m. on Thursday. So I'll double check that I have that as the due date, because I need to go over that lab on Thursday. You know, I have to kind of keep us on schedule, because eventually then we're going to have a lab exam that corresponds to those, or it's more like a lab quiz that corresponds to those labs, right? So I just want to kind of keep going over them as they're due, right? So let me just share my screen real quick, and then we're done. Again, I'll stay on if anyone has individual questions that they want to go over. I'll stay on for a few minutes just to make sure. But again, just a reminder, right? You have 2 assignments for that metric and microscope lab. The demos are up. They're videos of me in the microscope. There's probably another lab where I'll probably use the microscope in the lab maybe and have a little bit of a clearer view for you guys. But honestly, a lot of the microscope is you guys actually doing it. And so I don't want to focus too much on that, right? Because you even get to get the hands-on experience. But anyway, 2 assignments. You got this metric and microscope lab assignment, and then also the metric system lab assignment, which is super quick. It's just some conversions. All right. Homework chapters 1 through 3 is due now. It's already due. Like I said, the lab stuff is due on Thursday. The chapter 1-2 quiz is also due today. Okay. And then I will be uploading the recordings for chapters 4 through 6 in the next upcoming days. By Friday, all of those will be up. So then you want to start getting on, doing the next set of homework and quizzes. So there's a chapter 3-4 quiz, there's homework for chapters 4 through 6, and then be on the lookout for that next exam. Okay. And I'll also be putting up videos for the next set of labs as well. All right, guys. So that's pretty much it. If you guys have any other questions, I'm staying on. Just let me know. I did record this. I'll post the recording just like I did for the last ones as well. All right, guys. Take care.
Thank you.
Thank you.
Thanks, guys. Any questions, guys?